So today we will chat about the chemical methods, mostly, and some physical methods, for the control of microbial growth. Before we actually get into the nitty-gritty of those methods, let's define the major protocols. Okay? So the main protocols for the microbial control are separated into two large groups based on the nature of the surface you try to control the microbes on. One type of surfaces are fomites. Fomites are simply the inanimate objects, such as tables and chairs and computers and hospital beds and doorknobs and faucets, you can name it. So there are three protocols that are different in terms of the depth, the cleanliness, and the approach that is taken to control microorganisms. The most strict protocol is sterilization. Sterilization can be defined as the process in which all microorganisms, including endospores, are completely eliminated from the item. Sterilization can be achieved by using fairly aggressive physical methods, such as pressurized steam or autoclaving, or a set of fairly aggressive chemicals. A notch below sterilization is disinfection. So disinfection cannot guarantee the complete obliteration of all forms of microbial life, but it dramatically reduces microbial load on the fomite by physical, such as heat, radiation, or chemical methods. An example of disinfection would be the application of things like bleach or highly concentrated ethanol, which is a bit wasteful, okay? And then the lowest degree of the microbial control on the inanimate surfaces is sanitization, which reduces the loads mostly, and I want to emphasize it, so I sort of disagree with that statement, mostly by the mechanical way. So you effectively sanitize your dishes when you wash them. Does the dish soap do a lot? Not really. It helps you to remove microorganisms mechanically with the flow of water. That make sense? Now, the removal of microorganisms from the living tissue poses a certain amount of challenge. Okay? And the challenge is that, living tissue is pretty sensitive. So there are two protocols that are associated with the living tissue: antisepsis and degerming, or degermation. Antisepsis is effectively a chemical way to kill microorganisms. Okay? So, see antimicrobial chemical here? So application of hand sanitizer, application of chlorhexidine, scrubbing into the surgery, okay? Scrubbing using betadine or chlorhexidine, all of that is examples of antisepsis. Degerming is the reduction of microbial load by scrubbing. When you wash your hands with the regular soap, soap helps to remove microorganisms from the surface of your skin simply by scrubbing your skin and just wash them away. Does that make sense? At the same time, if you apply alcohol sanitizer on your skin, alcohol kills microbial cells because of its chemical properties. That's going to be antiseptic treatment. Okay, make sense? Now, Obviously, it's always better to prevent microorganisms from getting free than to fight them when they are established themselves in the environment. So in order to prevent microorganisms from spreading, we have containment levels. I'm going to start with the BSL-1 containment level. This is the containment level we are working at. So microorganisms do not cause, I will specify, severe disease in a healthy person. Okay? Have minimal risk to the environment. And even if they do have a capacity to cause the disease is very easily treatable and not life-threatening. Okay? So, in the tables here, you have, for instance, E. coli. I think you even have E. coli that is a broth. Okay, I don't remember. I think, yeah. What's going to happen to you if you drink that entire broth of E. coli? Huh? So again? More specifically, like, what's going to happen? Are you going to die? No. You're going to sit in the shitter for 3 days. And then you're going to be fine. So, like, it's not life-threatening, really. Like, it's going to be uncomfortable, but not anything deadly. Make sense? Huh? Well, if kids are drinking E. coli in this room, we have much more serious problems. And I probably should be fired or put in jail, you know? Kids are not allowed here. Okay? But yeah. And you bringing up a great point. Different people will respond differently, but we can't rely on certain outliers that, you know. Like, we can't judge the entire population based, for instance, on immunocompromised individuals, because they're not the majority of the population. And BSL-2, sorry about that. 7 here. BSL-2. So microbes are indigenous. What does that mean? Indigenous microbes are present normally in the population at certain locale. Okay, so for instance, influenza type A, which circulates right now, is an indigenous microorganism. Like, it's fine. You can have it in the hive. Okay? Disease is not severe. Does it make sense? So, influenza type A will be a BSL-2, maybe BSL-2 plus pathogen, because if you get infected, you're going to die. Probably not. Can you get a vaccine? Yes. Can you get a treatment? Also, yes. So, BSL-2. BSL-3. So to give you an idea of what BSL-2 is, there is a biosafety hood right there behind Isha. When you work with BSL-2 microorganisms, you have to work with them under the BSL-2 conditions, literally in that hood. Okay? It provides the barrier between you and the microbes. Okay? There are certain rules on containment of the waste, like you have to put it in the bleach and this and that and autoclave and whatnot, but it's all pretty straightforward. Okay? Now, when you work at BSL-2 conditions, do you have to wear a lab coat? Only if you want to look cool. Do you have to be 18? Yes. Yes. According to the US law, the person who is under 18, they aren't allowed in the room that is categorized as BSL-2. Okay? Then comes BSL-3. Yours truly actually has plenty of experience working in those conditions. So, first of all, which microorganisms? Microorganisms can be indigenous, say SARS-CoV-2, which is present in the US, or they can be exotic. For instance, yellow fever virus, which I worked on, which is not normally present in the United States. Okay? They can cause serious disease, potentially lethal, not necessarily only through respiratory transmission, but including respiratory. Okay? So, from my own experience, how that looks. You walk into the so-called ante room, the prep room. You put on a Tyvek suit with a hood, and then you put shoe covers, and you put a special filter with like a helmet on your head, okay, which filters the air to prevent respiratory transmission. You have to be properly trained. When you bring something in the BSL-3, you cannot take it out. It's considered to be contaminated. You have to go through the FBI background check and a bunch of other stuff, okay? So it sounds really awesome, like science and stuff, but it really is a pain in the ass because you sweat like a pig and everything is very cramped. And then there's a BSL, and I already give you examples of microorganisms that you would work with under BSL-3 conditions. Yellow fever, Brucella abortus, plague, you can treat them. They're dangerous, but you can treat them or you can vaccinate against them. So if you do all the precautions, you're safe. And there's BSL-4. Microbes are exotic, so not indigenous, not present. Okay. High risk of infection. I'm leaving out aerosol transmitted. It all depends. Frequently fatal. So, in the continental United States, I can name probably all of BSL-4 facilities right now. There is one in Fort Detrick, Maryland, at Walter Reed Army Research Institute. There's one at NIH there as well, so 2. There's one in Boston, that is 3. One in Atlanta, it's 4. One in Fort Collins, Colorado, it's 5. There should be 7. One at University of Texas Medical Branch, that is 6. And one at Rocky Mountain Labs in Helena, Montana, that's 7. So U.S., which is a powerhouse from the scientific standpoint, has 7 BSL-4 facilities. If you want to actual peek into what it looks like, this link, I think, still works. This is a movie that was shot by a bunch of virologists that went into still undeployed BSL-4 facility in Boston. Okay? So undeployed, the biologists called it cold, meaning that there is no virus work. When there is a work, it's called a hot facility, okay? So you can see how it looks like in a nutshell. Extreme security. When you go in the lab, you strip down to your underpants, I'm not exaggerating, putting sterile scrubs, and then going into a rubber suit that completely separates you from the environment. The only way you receive air is through the pipe that is sealed to your suit. Okay? And you can switch the pipes, but you have absolutely zero physical contact with the environment. Only suit does. I cannot imagine how hard it is to work in such conditions. It really is. I really want to do that. It will never happen. Because there are no visitors. You cannot visit BSL-4. Okay? So that brings us to a virus that is really overstated. BSL-4 pathogen, Ebola virus. Okay? So what causes Ebola virus infection? It's the enveloped RNA virus. Okay? Transmission. Initially, it is transmitted from animals to humans. The main idea is that the reservoir, the animals that carry that virus are bats, but it has never been confirmed. Okay? humans to humans transmitted with the contact with the bodily fluids, such as vomitus or urine or sweat or feces, okay? There are recorded cases of sexual transmission. Apparently, virus can stay in the testicles and just be sort of like chilling in the testicles, and then when a man has sex with a woman, the virus is transmitted with the semen. Cases are in the single digits, okay? It's not super common. Ebola virus is never transmitted by the respiratory route, oral route, or by vectors such as mosquitoes. Symptomatically, this virus causes hemorrhagic fever. If you will Google hemorrhagic fever, one of the first hits in images is the person with the blood coming out of a cephalic orifices, let's put it this way, ears, eyes, and nose. This is a complete and entire bullshit. Hemorrhages happen internally. They result in the leakage of plasma out of the blood vessels, and people go into a shock. Does that make sense? Hypovolemic shock. It's been demonstrated that antipyretics that will reduce body temperature and supplementation of the IV fluids can drop the mortality from typical 70% to 10%, which is still horrible, but it's not 70. Okay? At the end of the day, people who cannot receive any proper treatment develop organ failure and hypovolemic shock, as I mentioned. So case fatality rate, what does that mean? Let's say we identified that we had 10,000 cases, confirmed cases of Ebola, 5,000 people died. So half of them, it's 50%. Now the actual mortality rate does not take into account people who died of Ebola but were never diagnosed, and people who had Ebola virus infection but were never symptomatic. There are such examples. So if you look at the most famous outbreak of 2014-2015, which was largely confined to these Western African countries, Sierra Leone, Liberia, and Guinea, a little bit of Cote d'Ivoire, okay? You will see that for about, 30,000 cases, and what's the difference between the confirmed and just a case? Confirmed case means that there was a lab result. But if it walks like a duck, it's a duck. So if a person has all the signs of a hemorrhagic fever, even without lab confirmation, you can fairly easily count it as Ebola. So you can see that in this large outbreak, the actual case fatality ratio was closer to 33%. Maybe even slightly more than that. That make sense, everybody? Okay. So this outbreak, as I mentioned, happened in these countries for the most part, Guinea, Sierra Leone, and Liberia. And why did it happen? So there's several reasons. Well, one of them is the public health system is on the brink, okay? Not enough money. Negligence by the government, and I mean the local governments, as well as, to be honest, WHO. I always tell my students that if there is a work, like a job, that I really, really want without any moral repercussions, it's to work for WHO. You get shitload of money and you have no responsibility. The chief of WHO, after the outbreak subsided, she acknowledged that they fucked up. And she kept her position. Nobody fired her, she didn't step down. Okay? The response was very much delayed. And there's a lot of the public issues with this. There is a general mistrust to government. So when people were showing up to bury the dead, the governmental agencies, the local folks were like, no, we're not going to let you because we don't trust you. Make sense? Some local customs were not particularly conducive to, limiting the outbreak. There was a famous case when some gentleman died of Ebola, and by the procedure, that person should have been buried separately. But family said, no, we're going to do the proper funeral. So the guy was in open casket, and like 100 relatives came by and kissed this guy who died of Ebola. You can only imagine the consequences of this, right? Now, interestingly enough, United States also had an outbreak of Ebola. It was in Reston, Virginia. Reston, Virginia houses one of the largest monkey research centers, okay? The primate research centers. And they had a shipment of monkeys. Monkeys had hemorrhagic fever, they were dying. None of the personnel got sick, but then they tested the personnel, and turns out they all got infected. They were just asymptomatic. It was a pretty mild Reston strain of Ebola virus. Okay? Now, during that 2014-2015 outbreak, the U.S. did not have, well, okay, let me hold my horses. U.S. did have 2 cases of Ebola virus infection that originated on the U.S. soil. So the first person on the U.S. soil who had Ebola was a guy with a lot of relatives in Liberia, which he visited, and then he came back to Dallas and felt sick, went to the ER, said, I feel shitty. The physician goes, did you go to Liberia? And he goes, yeah. Did you meet anyone or got in contact with anyone who had Ebola? And he goes, no, which was a lie because he did and he knew. So physician says, also like smart move, right? The guy was in Liberia, just, connect. But he goes, nah, it's fine. Go home. Next day, he comes back. He's in a much shittier place. Long story short, he dies soon because it's just too advanced. Okay? 2 nurses, they cared for him. Both get infected, which turns out later. They get infected. One of them, while infected, travels from Dallas to Cleveland and back. So they had to test both planes. Nobody got sick. Ebola is not super transmissible. Both nurses got treated, got much better, recovered fully. There were other cases, importation to New York. A few people were flown to Atlanta and Frederick. That's, CDC and Walter Reed Army Institute. There was one guy from Liberia, really sad story. He was a physician, American physician from Liberia originally, went to Liberia to volunteer to help, got infected, got evac'd to Omaha, but didn't make it. He arrived to Omaha, but it was too late. Okay? So, the outbreak of a disease like Ebola, despite, I know, we've all gone through COVID, but different story. The outbreak of disease like Ebola is not possible in Europe or US. We really have a robust monitoring system, and we have a robust hospital system no matter what happens in the country now. Okay? And Ebola is transmitted only when people are symptomatic. So when you can be infected, be in the incubation period, have no symptoms, you do not transmit this virus to anybody else. So this is why it's not. Now, I don't know, it was 2014. Some of you, many of you were probably in the school back then. Yeah? Those of you who weren't in school, do you remember the hype around the Ebola outbreak? Yeah? This is a really old data. It's table on the screen. Shows you annual deaths on the African continent from various causes. I want to show this. Let's say it's better, which I'm not sure, but let's put it 250,000 people die of protein energy malnutrition, also known as kwashiorkor, or you can call it starvation. That Ebola outbreak killed 11,000. To be honest, we should be up in arms about this, not Ebola. Okay? Does that make sense? But this does not provide really good leads and titles on the articles, you know? Can we prevent Ebola? Yes. There's a vaccine. I always ask these questions to my students. So, Ebola is happening on which continent? Africa. Now, to be straight, is it the richest continent in the world? No. Also, not a lot of people get sick. So it's not like vaccinating people against polio, right? So it's not really a commercially viable project. So obviously, African countries could not pay for the research. So the research was sponsored by one U.S. government agency. Remember, Ebola virus is not present in the U.S. Which government agency would pay a shitload of money to make a vaccine against the virus that is not in the United States? Nope. CDC does not have a shitload of money. Huh? Dude, these guys are an equivalent of a homeless person now. The agency that has a shitload of money. Yes, Pentagon paid. Because who knows where we're going to put democracy on the ground next, right? So we have to be prepared for all the challenges. They did pay it when the shit hit the fan. They distributed it, and it's not a vaccination of everybody, so-called ring vaccination. So let's say tomorrow they realize I have Ebola. I'm not getting vaccinated. You're getting vaccinated. Because you are my ring of contacts. Does that make sense? It's pretty good. Like, it really is good vaccine. We just don't have enough data to, like, license it fully because there are not so many cases. Can you prevent it? Well, treat it, yes. There's an antibody here, ZMapp, which, like, these top lines are the monkeys that received antibodies, and the black line is the monkeys that didn't. The monkeys that didn't die. Okay? Now, the problem again is the price. Monoclonal antibodies are very expensive. Right now, there is an outbreak of Ebola virus in Democratic Republic of Congo. It goes down, but it was pretty much up. A few thousand cases. Okay? Well, good. Now, this part here, I use to bring up, you know, the case for how different factors affect the survival of a microorganism. So, first of all, I want you to look at the Exhibit A. Okay? Which is sort of duh. You see that? If you put virus in the warm water, it gets inactivated quicker than if you put it in the cold water. Which is like, yeah, sure. This is one of my absolute favorites. You see these two lines? Yeah? Versus these two lines. So the lines that I highlighted here, these represent the behavior of the virus in the drying blood versus the behavior of the virus in the liquid blood. When you put a microorganism in a bodily fluid, it becomes very much protected. I'm telling you this because when you're going to be in the hospital, it's not like your patients spit out or cough out or shit out a pure virus or pure bacteria. They are entrapped in mucus or blood or whatever, right? And those bodily fluids really protect that stuff. So, like thinking, oh, I'm just going to spray it and leave it for 15 seconds. No way. Because that blood will sort of take some of that inactivation action on in itself. Okay? This is nothing surprising. So I love those types of studies. They're so elegant. So what they did, they checked the sensitivity of Ebola virus to different antimicrobial chemicals. These are good. Destroy it in 5 minutes. So which two suck? Well, very much diluted sodium hypochlorite, which is bleach. If my memory serves me well, the bleach concentrate that you get from the grocery store is this blue one, 0.5. Okay? So if it is of any moral relief, the concentrated bleach will kill Ebola virus. You're safe from Ebola virus in your house. Okay? But if you dilute it, it's not going to be as effective. But look at this. 67% ethanol. So basically hand sanitizer. I have a question for you. If hand sanitizer and bleach kill Ebola virus fairly equally, how come if I ask you, like, I'm gonna pay you $1,000. You have to put for like 10 seconds in your mouth. You don't have to swallow. 10 seconds in your mouth. Bleach or hand sanitizer? What's your choice? Yeah. So you perceive it as safer, right? Which it is. How come that it's as effective against Ebola virus as the bleach? You don't have to answer that question. I'm gonna keep the suspense because we're gonna answer it later. But there is a pretty good scientific explanation to this. What I seriously don't understand is this graph. It makes no sense. This is data, but I can't wrap my mind around it. So if you look at this data, the virus can persist for a pretty long time on the respiratory mask, which I'm fine with. It's porous, it can hide in there. But the fact that it gets quickly inactivated on the cotton gown, basically a big-ass mask that covers your entire body, makes no sense to me. Okay? I would expect that any porous material will be more protective compared to the steel when the virus is exposed to, all the elements, so to say. But nevertheless, the data are data. Okay? So let's talk about how we control microorganisms, and we will start with sort of general aspects. So you can see this list here in terms of sensitivity to control methods. So in this specific sequence, we go from most resistant to least resistant. In other words, endospores are very hard to kill, and enveloped viruses are not. I will ask you to pick the most or the least resistant organism out of the list on the exam. Am I clear? You may wonder, what if I ask about the least resistant organism and put trophozoites and enveloped viruses as two options? I won't do that. Does it make sense? I will make it unambiguous. Okay? How can we quantitatively characterize the effectiveness of the method? We can do it by the decimal reduction time. So what is decimal reduction time? So this is the death curve. Okay? Now this point right here corresponds to 1 million bacterial cells. Do you agree with me? Yeah? And then this point corresponds to 100,000 bacterial cells, which is 10 times less. And how much time did it take to reduce the population from 1 million to 100,000? It took us 5 minutes. So for that particular microorganism, whatever it is, that particular method, the decimal reduction time is 5 minutes. Okay? Make sense? Now, if I change temperature, or pH, or, you name it, it will change. So it's always for a specific condition. And as you can imagine, the lower value, the shorter period that's required to get the population of microorganisms down 10 times, the more effective the method is. And the longer, the less effective. Okay? Now, methods that we apply can be cidal and can be static. Cidal means that they kill. Static means that they just stop microorganisms from growing. And the first group of methods are the temperature-based. First rule: moist heat is more effective than dry heat at the same temperature. Make sense? The resistance pattern is the same, but what did I mean by saying that moist heat is more effective than dry heat? So what you see on this graph are the death curves of Bacillus subtilis using dry heat. So time is on the y-axis and the number of cells in the x. So dry heat versus moist heat. Look, same temperature. Now, how much time did it take for moist heat to reduce the number of microorganisms tenfold? 32 minutes. Look at the dry heat. 415 hours. Okay, so it's like 800 times more. Okay, does that make sense? So it's a very big difference. Okay, now keep in mind, Bacillus subtilis forms endospores. So vegetative cells were gone pretty quickly. It's endospores that they were frying for, 15 days or something. Okay? Now, two more parameters. Thermal death time. Okay? So the time that's required to kill, all the microbes at a certain temperature. And thermal death point, which is the lowest temperature that can kill all the microbes in the sample in the 10 minutes. Okay? No definitions. So this, if you are confused, you have all the rights to be. So I was very excited when I made this slide, and then now I'm very disappointed in myself. So here's what you need to know. Let me highlight, and I'm gonna write on this to avoid any ambiguity. First, what you need to know. For a method like boiling, you should be able to tell me what it is. Well, obviously not boiling, but like autoclave. What is autoclaving? Pressurized steam, okay? And you must know what level of control, it can achieve. Am I making sense? So, boiling, disinfection. It is not sporicidal. It does not destroy endospores. Okay? So, dry heat. Can sterilize. You're going to spend a lot of time there. If you want sterile, we're talking days. Okay? Incineration, which is burning. Do you think it's going to destroy everything? Yeah. So definitely sterilization. Autoclaving. Sterilization, very easy. Do you need to memorize any settings, like the specific temperatures or pressures? No. Okay. Pasteurization, pedantically, this is disinfection level. Does anyone drink unpasteurized milk? Good decision. Huh? Bad decision. Campylobacter jejuni, gastroenteritis. E. coli, anything from UTI to diarrhea to dysentery. Salmonella, I had a mild form, still remember, when I was 9. I had a mild salmonellosis. I thought I will see my stomach finally falling out of my mouth. Yersinia paratuberculosis, severe diarrhea. Listeria monocytogenesis, extreme diarrhea, sepsis, neonatal meningitis, and miscarriages in pregnant women. All of them come from those cute cows. Okay? There is a reason why we do that.
So that would be your
Unpasteurized milk. Now, if you think that, if you like the eggs sunny side up, it's a bit of Russian roulette. It is. Okay? So, my point is pasteurization neutralizes all those bad actors, you know? Now, what about this? So, refrigeration and freezing, we consider both of them microbistatic. Which means, if I have a rotten piece of meat, I throw it in the freezer, and I take it out, it will keep rotting, because you don't kill all the bacteria by freezing. Does that make sense? There will be no questions on this. 2 reasons. Oxygen therapy is more of a medical treatment, specifically for anaerobes. High pressure processing, I read the papers. It is not impressive. I mean, the effectiveness. I really read the papers, the research papers. It's not that impressive. Desiccation, also static. Okay? So, drying up, great bacteriostatic treatment. beef jerky, an example. Okay. Well, we talked about salt. Lyophilization actually is a method of preservation of bacteria. You freeze dry them, and then you can reconstitute them back. Okay. So in a nutshell, right now you have these most important methods right here. Okay, to understand what level of control they provide and what they are. Now, what about radiation? Radiation is great. There are two types. So, ionizing radiation, X-rays or gamma rays. Okay, does that make sense, everybody? Let me make it a little thicker. Oh yeah, that's better. What level of control does it provide? Sterilizes. Now, here's the point. Let's say I want to autoclave my mug. Should I? Would you be concerned about any part of the mug being damaged? Which one? The plastic, the lid, right? So, the metal is going to be fine, but the plastic is not. So imagine I have like a plastic something that I need to sterilize. There is a plastic that withstands sterilization in autoclave, but most doesn't. So what do? I throw it into the gamma ray chamber. So you have, like, it is a conveyor belt. There's the chamber that the material, whatever things go through, and then there's a source of gamma radiation that constantly radiates them, so they receive the dose and they sterile. This is how lab plastic is being sterilized. Okay? Gamma ray treatment is also used to treat fruits and vegetables. This is the label for radiation treated produce. First question, why? Why treat with radiation? Huh? Shelf life. Simply shelf life. It very much increases shelf life. Okay? There is another reason, though. This is a very specific story, which I find very endearing. United States import mangoes, usually from Central and South America. Turns out that we are not the only mango lovers. There are flies that deposit the eggs in the skin of mangoes, and they develop into larvae. Okay? So there were cases described when a person ate a mango down south in south, like the mango that was taken from a tree, literally, like, and given, and the woman ate it, and next morning found little white worms in the feces. She freaked out, of course. So that was larvae of those flies. They are absolutely harmless for humans. They probably die in our digestive tract. But again, imagine this will happen to somebody in the state of Ohio, right? So mangoes are irradiated, so all the eggs are killed. Okay? So you're still going to eat those eggs, but they're not going to reveal anything. Okay? So food sterilization, plastic sterilization. No, it's with radiation. They say it's sterilization. No. Say again? Yeah, X-ray would have the same effect. Just 1 second, folks. So, X-rays, yeah, can be used, totally.
How does it not, like, does it like, cause cancer, though?
Nope.
Like,
Nope, because that's the waves. So they kill bacteria, but that's it. They do not induce any residual radiation. So no, it's totally fine. Yes, so non-ionizing radiation, specifically UV light. That's disinfection. But
Is gamma radiation preferred over X-rays?
I think whatever is cheaper,
Okay.
whatever people can build, like seriously, it's the main concern. Okay, so hold on. Yeah, okay, perfect. So the reason why gamma rays are better, they have greater penetrating power. Gamma rays are the most penetrating, so they can go through things. Then X-rays, UV light is not really penetrating. Okay, doesn't go through the door. So usage, air disinfection, dental offices, for example, that biosafety hood. After I'm done, if I would work there, after I'm done, I would turn on the UV light to disinfect the surfaces. And if you are filthy rich, like the royal families on the Arabian Peninsula, you can build pools in hotels that are filled with the ocean water, but it's disinfected by the UV. It costs an enormous amount of money, but they can afford that. Okay? So, just a quick reminder, UV lights, how they affect bacteria. They introduce diamond dimers in the DNA, and gamma rays just break bacteria in pieces. Now, the last of the physical methods that we're going to chat about is filtration, and this is the only method to do anything about gas. Seriously, like if you need to control the quality of air, filtration is the only answer. And it also is very important for very specific sterilization procedures of the fluids. So you can pass the gas through a membrane, which will filter out particles. So, HEPA, high efficiency particulate air. So air filtration can achieve different degrees. Ultimate question, can you sterilize air? The answer is yes. It's extremely expensive. Okay? For biosafety level 4 facilities, where people work with, viruses and stuff, the largest, article in the budget is air handling. Energy that is associated with filtering air is the biggest money sucker in those facilities. Okay? Now, why do we like to use filtration for liquids? The answer is that they can contain heat-sensitive components. If you will try to sterilize some kind of a drug, you may very easily degrade it because of the temperature, right? Sterilized by autoclaving. Do you follow my train of thought? But if you filter it, filter will block the passage of things like, viruses and bacteria. But all the chemicals will go through and you are safe. Does that make sense? That is actually, this is how temperature... I worked at the manufacturing facility for various biologicals. This is how they sterilized. It's filtration. It's never... it is never autoclave. Because the composition must remain the same. And filtration keeps the composition. Okay, we're good so far? Now, chemical methods of control. This is going to be more sort of relevant for you because we're going to talk about the stuff that we all use at home. Okay? So in general, what do you want? Works fast, low concentrations, can resist inactivation by bodily fluids, non-toxic, kills everything, penetrates the surface because if the surface is porous, you want it to get in. Doesn't corrode things, doesn't stain things, smells good, affordable. This is Prince Charming, really. You know, it doesn't exist. So the general rule for chemicals is that the better it is at controlling microbes, the more toxic, aggressive, or corrosive it is. Okay, and I will show it to you in multiple examples. So when you choose the agent that you need, okay, so nature of microorganisms. Now as you may have noticed, I love to bring data to the table. So look at this. Different chemicals were tested in their ability to inactivate common microorganisms. Staph aureus, wound infection, salmonella, E. coli. This is the nasty strain of 157H7 that causes severe diarrhea and hemolytic uremic syndrome. Pseudomonas, really bad respiratory pathogen. So you can see that a lot of stuff that we use, like Vespine is some kind of a phenolic compound, TBQ, something like kinda Lysol. Clorox doesn't need introduction, ethanol, Lysols, Mr. Clean, vinegar, and then soda. So, how many of you heard like some influencers saying that vinegar works as antibacterial? It's bullshit. It does not. Like, you may, if you wash fruits with vinegar, you may wash it with the piss. It's pretty much the same effect, and baking soda even worse. Okay? There's nothing. There's no reduction. Okay? So also, notice I have some unhealthy negative attachment to Lysol disinfectant. I kind of want this thing off the shelves. Out of all actual chemical disinfectants, it sucks. It has the lowest efficiency. Okay? So, we actually test Lysol, I don't think here, but at Lakeland we do. And it is the lowest scoring antibacterial chemical that you can buy in the store. Question now?
If baking soda doesn't reduce a lot of bacteria, why do they tell you to use it after you get your maybe it's just a wives' tale to use it to get rid of a canker sore or something?
What is a canker sore?
Oh, like an ulcer in your mouth.
No. The only thing it will help with, your mouth is generally basic. Saliva has pH over 7. When you eat something, lactobacilli start growing, make it acidic. So to kind of bring pH back, but it does not have any significant effect on bacteria in the sore or on the teeth. Okay, it's not really antibacterial. Okay? What? Can't I?
Would using salt be better than using baking soda then?
You have to hold it in your mouth. Salt, you have to hold it in your mouth for a long period of time. And it's fine, but remember that you're not exclusively killing bacteria. You're also sucking the almost literally living juices out of your own cells. No, seriously, it's a hypertonic environment for them as well. It's not like they are whatever, So, okay. Now, for chemicals, things to consider. And by the way, selection. I kind of steered away. Okay. Well, nature of treated material, obviously. Degree of contamination, sure. Time of exposure and microbicidal promises. But here's another thing. I'm going to give you an example from our own lab practice when I was postdoc. We had to spray cages with mice. when we move them around. Like you bring it to the lab, if you want to move the empty cage to like storage, you have to spray it. And we had bleach. It was mandatory to spray them with bleach. And we moved the cages on the carts. You know these metal carts? After we went through 2 carts in 4 months, because bleach corroded the hell out of them, we had to switch to plastic. So when people select the agent, you kind of have to think, is that a good choice for that specific purpose? Okay? Now, I'm going to give you some, remind me to give you a summary about the questions on this topic. Okay? So halogens. Out of all halogens that there are, we use chlorine and iodine. All of them work through oxidation. That's their mechanism that you should know. So chlorine and iodine used for 2 very different purposes. Chlorine is disinfectant. Okay? If you leave concentrated bleach on the surface for a long time, it will start killing endospores. This is hard. I mean, to leave it for a long time, because you probably need those surfaces. Like if it's your kitchen counter, unless you bleach it overnight, that's not sporicidal. That's disinfecting, okay? For the most part though, chlorine, if you don't mind the smell, is one of the best, if one of the best disinfecting solutions you can think of, okay? You know that chlorine is used for water treatment. Now iodine, also oxidant, but it stains. Make sense? And it's a bit of a pain in the ass to make a solution of iodine. So we can't really use it for disinfecting surfaces, but we can use it for aseptic techniques. So you're familiar with betadine, right? The brown stuff that they use on your skin, yes? Yeah, that's iodine solution. Okay. This data, like this link that I give you, this is the data from it. You can see that it takes like less than a minute to destroy all coliforms, fecal bacteria. Okay, alcohols. I'm going to circle back to Ebola now. So we use 2, ethanol and isopropanol. We don't use methanol. Anyone knows why? So when I was in the lab, we had a flask with ethanol, and the label on that flask was buffer C. Why did we not put the name on the flask of the actual ethanol? Yes, because people would drink it. And people would drink anything that smells like booze. Methanol, ethanol, and isopropanol smell like booze. The problem is, methanol is extremely toxic. If people drink, and they do, hand sanitizer that contains ethanol, they're going to get drunk, they're going to fuck up their liver, but they're not going to die. If they're going to drink isopropanol, they're going to go into ketosis, call 911, and people will save them most likely. If they drink methanol, by the time they realize they poisoned themselves, they're on the route to probably hell. Okay? Does that make sense? So this is why we use those. How do they work? They disrupt the membrane, the nature protein, and suck the water out of the cells. Okay? They disinfectants, and this is the moment when we circle back to Ebola virus, to some extent. So the alcohols are very effective against enveloped viruses. Do you see the table on the right? Yeah? So, influenza and Zika virus are enveloped. Now, the highlights that I already have in place show how much ethanol, well, not different chemicals like water, Mountain Dew, which I find hilarious, neutral red dye, and ethanol, 70% ethanol. How much they... Okay, how much ethanol decreased the activity of the virus, right? So if you look at Zika and influenza, that's pretty substantial. That's like 6 logs, 5 logs, and then you start looking at the naked viruses. And for them, the numbers are much more noticed, okay? So here's the deal. Enveloped viruses have the envelope, which consists of phospholipids, just like cell membrane, right? Alcohols are great at destroying the envelope and inactivating a virus. Naked viruses do not have a membrane and not sensitive to alcohols. So remember how alcohol was as efficient as the bleach against Ebola? Ebola virus is the enveloped one. Okay? Now, I'm trying to give you connections. Anyone works in healthcare? I was expecting that. Do you wash your hands? Do you use hand sanitizer? So if you dealing with a person who has COVID or flu, will hand sanitizer help? 100% yes. Influenza and SARS-CoV-2 are enveloped viruses. If they end up on your hands, you apply, leave it for really like few minutes, like let it dry out. All the viruses are inactivated. You have a child, say 6 months old, who has norovirus. Are you familiar with noro? Yeah, 2-bucket disease. Yeah, you need 2 buckets. So you're dealing with a child, and is the table there? Okay, with the norovirus. You apply hand sanitizer. Is it going to work? No, because it's a naked virus. Okay, noroviruses, enteroviruses, adenoviruses, all naked. Rhinoviruses, that's common cold, can only be removed by washing your hands. So combine 2 methods.
So naked viruses would require.
Naked viruses are generally way more resistant than enveloped. There was a study, I think the most resistant human virus is papillomavirus. The study showed that you need 1.5% bleach, which is like industrial grade, and it's some considerable time to inactivate. Like, it's seriously resistant. Okay? All right. Peroxides, that's oxidation. Reactive oxygen species. Can be sporicidal at the higher concentrations. Okay? So, vaporized hydrogen peroxide is used to sterilize rooms. You just put a tank, turn it on, walk out of the room, seal it, turn off HVAC. The room is filled with H2O2 vapors. Everything dies. If you don't walk out, you die as well. You can mix acetic acid and hydrogen peroxide and create peracetic acid. Don't do it at home. Peracetic acid is really a great, fantastic sterilizer. It's also very toxic. The vapors, seriously toxic. My wife worked with peracetic acid for a couple of years. She would work on Friday and would have trouble breathing the entire weekend. It's bad. Surfactants. So, in your house, the cleaning agents that you use, most of them are quaternary ammonia compounds. Like if you take those Clorox wipes, it's actually not chlorine. Okay? So how do they work? If you look at this guy, acetylpyridinium, it has a positively charged head with the nitrogen atom that has 4 bonds. I'm not going to ask about 4 bonds, don't worry. This is what I refer to when I say quaternary ammonia. And then you have this really, really long tail, which is hydrophobic. So you have hydrophilic, okay, part, and you have hydrophobic part. What is the charge on the phosphate head of phospholipid? It's hydrophobic, but what is the charge? Positive or negative? Phosphate is negative. So effectively the surface of the cell is negative. Okay, the surface of the membrane. Now, this guy, acetylpyridinium, is positive. So positive head of acetylpyridinium binds to the negative phosphates, then the hydrophobic tail gets immersed into the hydrophobic part of the membrane, disrupts it, the membrane is disrupted, cell lysis. That makes sense? So this is truly antibacterial chemical. Now what about N-ionic compounds such as soap? They have a negatively charged head. Can you see that? Negative soap and negative membrane will repel each other. So it works just purely as a surfactant to remove dirt and bacteria from your skin. While the quaternary ammonia, the cationic compounds, actually better control. This is not the greatest illustration, this graph, but basically this is cationic, and you see it goes up faster, and this is anionic, and it takes greater concentration to start killing. Okay? Phenols. To my knowledge, and you can imagine it's a nerdy pleasure to read the composition of different chemicals. To my knowledge, the only household chemical that contains phenols is pine sol. Okay? It contains phenols and quaternary ammonia compounds. Phenols were the first antiseptic chemicals. That's carbolic acid. They work through disruption of the membranes and denaturation of proteins. Bisphenols, like these guys and these guys, were used in the plastic bottles as antibacterial compounds. Turns out they screw up human endocrine system. So if you have a plastic drinking bottle around you and you look at the bottom and it says BPA free, that's it. And then there is a story of triclosan. So it was an antimicrobial compound that was developed, I don't remember which company. They tested it and it looked really good in tests, and the concentrations that they used, it was safe for humans. So they started adding it to soap. Turns out, being a component of soap, it wasn't that great. And on top of that, all that triclosan went into the groundwater. What happened? How did bacteria in the groundwater change when triclosan started leaking in massive amounts in the environment? Yes. Bacteria very quickly developed resistance to triclosan. And FDA, I believe in 2018, officially announced that we say, don't use it anymore. So basically, if you see somewhere it says antibacterial soap, it's most likely a scam. Okay. chlorhexidine. Does it sound familiar?
Yes.
One of the best antiseptics that we have. It's disinfection antisepsis, but it's very safe, it's very mild, and it's very effective. That's all I can say about it. Okay, but it's great. For instance, there were studies showing that it is not inferior to iodine when somebody scrubs it. Heavy metals. Out of these 5, we actually use only 3 in the center are useful. So we used mercury, the very first one, for the ceilings, the dental ceilings, but it's not a great idea. Mercury is very poisonous. Zinc. Are you familiar with all those drugs like zinc for the colds? Yeah? Yeah, it's a scam. There's no absolutely zero data, like multiple studies showing that they do not really do anything to respiratory infections. Okay, so just no. Now, copper, gold, and silver. All 3 of them are very antibacterial, but the copper is cheapest. That's the reason why we use copper. And this is the study of addition of copper to different surfaces in the hospital. I'm not going to read to you all the legends here. This is the actual link to the study. It's a great, very, very precise, very elegant study. The results speak for themselves. On the left are levels of microorganisms on the hospital equipment before copper. On the right, with copper. Yeah, that's it. Now, I've heard that Cleveland Clinic started a program of replacing all the faucets by the faucets that have copper in the alloy. I don't know how true that is, but that would reduce, just that would reduce the level of hospital-acquired infections very dramatically. Thank you for bringing me back on Earth, because we never talked how they work. Yes, on the surface, very small amounts of copper or gold or silver bind to bacterial enzymes and block them. And this is the feature. You see the term oligodynamic? They work in very small amounts. The advantage? Bacteria cannot become resistant to them. Because, like, if copper screws up the protein so much, there's no way you can adjust the protein without changing its function. So, like, these are really, you can't become resistant to a fire. This is sort of a chemical fire for bacteria. Okay, does that make sense? No? Yeah? Okay. So, again, that's how they work. And, yeah, the last bit is alkylating agents. This is not the chemistry class. I'm not asking you to memorize the formulas. What I'm trying to tell you is that you have the alteration of the structure. When you start adding this is an example of an alkyl group. This is an example of the alkyl group. So when you start adding those groups randomly and change the structure of DNA or protein, you can denature them and they become dysfunctional, and it's not good. Okay, for bacteria, right? So for this particular mechanism, so what you need to know, modification of DNA and proteins, it's alkylation, and that alkylating agents can be gases. So formaldehyde, ethylene oxide, and propylene oxide, they're gases. The ones that I didn't highlight, fluids, you should know that. So what would you use gases for? They are very aggressive, they're sterilizing. What would you use a gas like ethylene oxide or propylene oxide? They can sterilize. Yes. Huh? Louder. Yes, hospital area. Can you be a little bit more? What do you mean by area?
Operating room?
Sure, a room. A room, okay? They can be used to sterilize rooms. Specifically, ethylene and propylene oxide are used to sterilize very sensitive equipment. Again, like plastic, okay? That you, for some reason, cannot irradiate. They're really good at it. Glutaraldehyde, phthalaldehyde, beta-propione lactone, the fluids, mostly used to preserve specimens. Does that make sense? For instance, beta-propione lactone is used to inactivate viruses to prepare vaccines, for example. That makes sense? Now, it kind of brings my memory back when I was working in research. I once lived through decontamination of biosafety level 3. Every year, biosafety level 3 and 4 labs go through decontamination. They filled with formaldehyde. It stays there for 2 weeks, then sucked out by HVAC. And everything that was inside is now dead. Okay? When they tell you that, it's gone, it's not true. We had to work there to clean up and stuff. It's horrible. You can work for 2 hours, then you have a migraine. And then you have to come back next day. But the reason for decontamination, think about this, I'm talking about the safety. If I bring a pen in a biosafety level 3 lab, I must leave it there until we do decon, because then it's all clean, I can walk in and do stuff. Does that make sense? So the only reason I brought it up, and I talked about BSL-3 lab so much, when you hear the debates about the origin of SARS-CoV-2 pandemic, I worked at these facilities. People who work there take the safety and security measures very, very seriously. Selling an animal that has this infection at the market sounds as a much more likely reason for the introduction of the virus to humans. Okay? All right. We're going to